But yeah, my name is Terrence Hanscome. I'm really excited to be here and tell you a little bit about some of the work that I've been doing using Iso-Seq to study recombinant AAV expression. Let me get this up here. All right. So let's jump in. All right. So just a little basic background biology on the AAV. It's a really small virus. 20 nanometers is the capsid right here. And in the wild type form, it's got two different genes, which they actually express several different versions of these rep and cap proteins. And so cap involved in capsid production, and then rep involved in replication. Rep is a really interesting gene because it actually it forms a nuclease, but it can also form a helicase. And so in the wild type form, it is flanked by these 5' and 3' ITRs that Don did a great job in describing. And the whole genome is about 4.7 kb single-stranded DNA genome. And so scientists have found out that you can replace rep and cap with a payload and deliver a therapeutic benefit to a cell. And so those ITRs are really neat because they allow for this phenomenon called rolling hairpin replication. And so it goes kind of like this. And so if you have these palindromes at the end, they actually enable priming and synthesis, replication of the genome. And so you get a polymerase coming in and synthesizing all the way across, making this, you have this like large hairpin structure here. And then because you have these palindromes at the end, they actually fold back on each other again. And so you get synthesis going in the other direction. So it goes back and forth. And so until you get this like really long structure. And so I had mentioned rep before that can act as a nuclease. And so rep will bind at what's known as the TRS site or terminal resolution site. And so that will make a cut, and it'll actually bind covalently to the 5' end of the genome. And this enables packaging. And so what we see here is we have rep bound to the 5' end of the genome, and this actually co-localizes with some other rep multimers that are at the porthole of the capsid, which have an ATPase helicase function, which shuttle the AAV genome into the capsid in the 3' to 5' direction. After lysis and infection into another cell, the genome can exit the capsid in the same orientation, 3' to 5'. And so scientists have found way before us that you can replace the sequence in between the two ITRs and deliver a therapeutic effect. And so this has been great for gene therapies. And so Don already went through this slide, so I'm not going to really belabor this, but the gene therapy can enter the cell and express as an episome, so not integrating into the genomic DNA, but can express a corrected mRNA, which expresses the corrected protein. However, you can still have, in this scenario right here, you have a mutated gene which is expressing a mutated protein. And so with our gene editing therapies, this will enter the nucleus and recombine with the genomic DNA, thereby generating a functional gene and a functional mRNA and protein. And so while we can deliver a therapeutic benefit, we want to know, are we getting correct isoform expression? Because if we're putting these into patients, we want to make sure that all these isoforms are what we expect them to be. And so the one way to do this is to characterize with RNA-seq. And so this is just a really basic RNA-seq overview. Aziz did a really good job of talking about this, so again, I won't belabor this. But here we have two samples of interest that are represented by two different mice receiving two different therapies. So once you carry this experiment through, you transduce with the virus, and you bring that through to the endpoint, whatever weeks or months you have. You can isolate the RNAs and generate your cDNA library. And so I do that for Iso-Seq using the poly-A tail. You can also use gene-specific primers or random hexamers if you want. Then here in this scenario, they're doing short read technology. So this looks like an Illumina instrument. And so here you end up with all these short reads that have to be mapped to a reference. And so you're getting these little snapshots of what's actually there, and so this presents the problem that you're not getting these full-length reads, you're not getting these full-length isoforms. And so this is where PacBio comes in, and Lara already has shown the slide. So here I'll just be real quick. You have your gene, and you have these 4 different mRNA isoforms. And so if we take say this for short read technology, we take this particular read into account. We don't know if this is coming from this mRNA isoform or from this one. And so PacBio's solution with Iso-Seq and long read sequencing has really been a game changer and has enabled us to see all of these full-length isoforms. And it's also great for isoform discovery, as Aziz had mentioned. Okay, so just real quick, this is what we do. So we have our cDNA sample, and you can do DNA damage repair, end repair, A-tailing, adapter ligation, and then once you anneal the primer and bind the polymerase, you can load it into the SMRT cell, and you see it down here. The polymerase is bound to the bottom of the well and incorporating those nucleotides of the ZMW rather. And so, all right, the movie's working. Here you can just see the polymerase going around this, and after going around hundreds of times, you actually you can get a consensus read that you can feel confident in. And so we use this for our Iso-Seq. And so for us, I just want to go over some of the goals that we had for our in-house targeted Iso-Seq, is to have a full picture of our codon optimized isoforms for our recombinant AAV therapeutic variants, to be able to quantify each of these isoforms, if there are any, to improve our codon optimization computational models with some empirical data, and I'll show a little bit about that, and then to include this as a part of the R&D pipeline when we're choosing candidates to further develop. And this is sort of what we do at the bench. So, after you transduce something and you carry that experiment out, you can generate the total RNA, you extract it, and a thorough DNase treatment is pretty important for this. Then you can generate your cDNA library, first strand synthesis, second strand synthesis. There are various PRONXB purifications, as with any sequencing library prep. But then with this, we have our hybrid capture. And so we use these biotinnylated probes to capture all of our isoforms of interest. And then there's the secondary amplification, and you get your PacBio library prep. I've also included an uncaptured sample prep here, just in case you want to do that. We did that, and I'll show you some data from that, why we did that. We really wanted to see what sort of fold change we had with our capture. All right. And so when we were building this assay, we started off with some in vitro samples. And so these were naive primary human hepatocytes. And the gene that I was looking for was ApoE. And the reason why we chose ApoE is because it's very well characterized. It has this very straightforward isoform profile. There are 5 different isoforms. It's highly expressed in the liver. And it is not impacted by the MOI of our vector or the multiplicity of infection. This is basically just like how many capsids per cell there are. So ApoE is, it's involved in Alzheimer's, so there's been a lot of research into it. And then, so once we had this all set, we went into our experimental samples, and so these were some in vivo samples, murine hepatocytes that were transduced with our recombinant AAV test vector. And so our target gene was the test vector payload. And so I can't go into any more detail than that, unfortunately, but but for ApoE. And so this is sort of what we expected to see. And so here you can see in the liver, there are those 5 different isoforms right there, which are represented down here with these 4 different exons. And you can see that 3 out of 5 in the liver are pretty highly expressed, and you can see that probably better represented up here in these normalized read counts. And so you get these top 3 isoforms that are pretty highly transcribed, but then you get these other 2 that are pretty lowly transcribed. And so we thought that if we can pick these up, then we could be confident that our assay was pretty sensitive. And so we could feel good about that. And so now I'm going to show you some data on that. So here, this is from our naive primary human hepatocytes. And so we just have a captured versus an uncaptured sample that we ran. And so here we have ApoE, which has been captured. We had the probes for it. And then this is followed by HP or haptoglobin, and then albumin. And so over here in the uncaptured sample, we have albumin, which is most of the signal. Most of the reads are albumin. And you can see that haptoglobin is actually only the 5th most transcribed gene, it has the 5th most reads. And I bring this up because what we found was that haptoglobin shares 132-ish base pairs of high similarity with ApoE. And so, this is just something to be aware of when you're doing a reaction like this, is that you want to know, whether your target sequence of interest has any similarity to anything else that's transcribed in your model, just because you may pick that up. So that's just something to be aware of. And we did pick up ApoE in our uncaptured sample, so that was good. We saw that really significant fold change in the captured sample. And just another way to look at this, here we're looking at the ApoE locus up here, and we're looking at the captured versus the uncaptured sample, and the y-axis here is just number of reads, CCS reads. And so up here you can see in the captured sample, all 4 of those exons are well represented. And then in the uncaptured sample, it's really hard to see they are there, but much more lowly. And then here, looking at the albumin locus for that uncaptured sample, you can see that all those exons are represented here, whereas in the captured sample they're very, very lowly sequenced. And so this is a very highly specific enrichment. And so one more slide with the ApoE data. And so here's the ApoE data compared to 2 different references, Ref 1 and 2, which are from rAAV and GenCode respectively. And then here with the data, if this is all the same data up here in purple. And so if you just take a look at the y-axis, we got 0 to 1 million reads, 0 to 50,000 reads, and 0 to 5,000 reads. And so we're just zooming down. We're getting more granular here. So you can see that all of these different exons or isoforms are represented. And so we were confident that we had a pretty sensitive assay on our hands. And so now I'm going to show you some data from some of the test vectors that we had looked at. And so here we're looking at test vector 1 and test vector 2. And so test vector 1, in one of our computational models for predicted splice donors and acceptors, and only 1 out of 3 of them, it was predicted that there was going to be there's a pretty high confidence of splice donor and acceptor site. The other 2 did not predict this, but we ran this sample, and then we saw, sure enough, we have this splice donor and acceptor site that was acting about 10% of the time. And then we changed that. We altered just a few base pairs to remove that splice donor and acceptor site, and that was resolved. And so that was pretty cool for us to just get that empirical data that was backing up one of the predictive models that we were using. And so I just highlight this because to show the importance of backing up your predictive modeling with some empirical data. And so this is just another way to look at this. And so this is test vector 1. Where we did see that splice donor and acceptor site that was resolved in test vector 2. And so transcription is going from right to left here. And so here we have our first exon, the first intron where that gap is. And we have pretty consistent transcription all the way across. And then we see this cup here, this like little gap. And so this was that predicted splice donor and acceptor site that we saw that was resolved in that second test vector. And so this technique is just really powerful in being able to get really granular and detect these subtle changes. Okay, in summary, real quick talk. So Iso-Seq is a highly sensitive assay. It's able to detect rare events, cryptic splice sites, polyadenylation signals, and then infrequently transcribed isoforms. It's an excellent system for isoform discovery. And then, I just want to mention again, just to be aware of sequence similarities between your target gene and other transcripts. And thorough DNase treatments can cut down on picking up anything that you don't want to pick up. And then, computational models are strengthened with empirical data. So yeah, I just want to thank the organizers for having me here today. I want to thank Dawn for being a great mentor, and everybody in my lab, John, Jason, everyone who's helped with this, Samuel. Thank you for listening, and I will take any questions.
Hybrid capture. For hybrid capture, are you? I guess the design there is you only find what you're looking for. So do you use like a kind of library of hybrid capture oligos, or do you just pick like the exon that you know is going to be in all of them?
Yeah. So I use a library of probes. I have kind of landed on like 4x tiling, using like 100 nucleotide probes and just like kind of stack tiling them like that. It's worked out pretty well. Yeah. Thank you. Hey.
So for the isoform counts, I've noticed there are fractions. Did you normalize them? Were you using the phonology counts from Iso-Seq data? Earlier there was like we were looking at the low abundance of the ApoE. We're showing fractions and counts. Earlier, this one.
Oh, this. Yeah. So this is somebody else's data. Yeah. That's normalized, I think, to transcripts per million.
Okay. Yeah. Okay. And another question is, have you started looking at whether there's a correlation between the isoform abundance and measured protein expression?
Yeah, we're in the early stages of looking at that for one of our programs, and there definitely are some differences, but I think that would require a lot more experimentation. But yeah, it's a really cool avenue we want to look at. Anybody?
Thanks so much.
Cool. Thank you.